Hello, my name is Christopher Flores, and this is my proficiency on DNA extraction. The purpose of DNA extraction is to purify DNA. It is to separate the DNA from the rest of the cell, membranes, proteins, and other cellular components. Now, once the DNA is purified, scientists can study individual genes, sequence the entire genome, modify sections of DNA, and many more things. Now, I will show you the procedure on how to do a DNA extraction. First, we have to warm up the elution buffer to 70 Celsius. Then, to a new nuclease-free Eppendorf tube, we scrape a bacterial colony into the tube with a pipette tip, as I'm showing here in this video. Then, to your nucleus-free Eppendorf tube where you have your bacterial colony, you add 200 microliters of PBS and you thoroughly resuspend the bacterial cells by using your micropipette tip and by just pressing the plunger and then unpressing it and doing this various times to resuspend the bacterial cells. You resuspend them till you have something that looks like this where there are no visible lumps. You then add 5 microliters of lysozyme to the tube. Then you incubate the tube for 15 minutes at 37 degrees. Then add 200 microliters of binding buffer. Then add 40 microliters of reconstituted proteinase K. Then add these components together and incubate for 10 minutes at 70 Celsius. After incubating, add 100 microliters of isopropanol and mix. After all these previous steps, transfer the solution to a high pure filter tube. Centrifuge for 1 minute at 8000 G. Discard the flow through. Add 500 microliters of inhibitor buffer. Centrifuge for 1 minute at 8000 G. Discard the flow through. Add 500 microliters of washing buffer. Centrifuge for 1 minute at 8000 G. Discard the flow through. Centrifuge for 10 seconds at 13,000 G. Discard the collection tube and put the filter into new Eppendorf tube. Add 50 microliters of elution buffer that was warmed up to 70 Celsius. Incubate for 30 seconds. And finally, centrifuge for 1 minute at 8000 G. Here is a picture of an agarose gel with a successful DNA band. It should be noted that this is not the DNA band I got from the DNA that I extracted from this video. This is a picture that I got from a project that I did in class, but I just needed to show a successful an agarose gel with a successful DNA band, and here is one.